Epistasis is a genetic phenomenon where the expression of one gene is modified or masked by the expression of one or more other genes. This interaction between non-allelic genes leads to deviations from expected Mendelian inheritance patterns. It's a crucial concept in genetics because it highlights the intricate interconnectedness of genes in determining an organism's phenotype. 1. Types of epistasis. Epistasis is classified into several types depending on the nature of the interaction between the genes involved. These include recessive epistasis. A recessive allele at one locus masks the expression of alleles at a second locus. An example is coat color in Labrador retrievers, where the EE genotype masks the expression of the B allele responsible for black or brown fur. Dominant epistasis. A dominant allele at one locus masks the expression of alleles at a second locus. An example is fruit color in summer squash, where the dominant W allele masks the expression of the Y alleles responsible for yellow or green color. Duplicate recessive epistasis. Recessive alleles at either of two loci mask the expression of dominant alleles at both loci. An example is flower color in sweet peas, where recessive alleles at either the C or P locus result in white flowers. Duplicate dominant epistasis. A dominant allele at either of two loci masks the expression of recessive alleles at both loci. An example is on character in rice, where the presence of a dominant allele at either the A or B locus results in on rice. Dominant inhibitory epistasis. A dominant allele at one locus inhibits the expression of both dominant and recessive alleles at a second locus. Polymeric epistasis. Dominant alleles at two or more loci act synergistically to produce a unique phenotype with a greater effect than would be produced individually. 2. Causes and mechanisms. Epistasis arises from various molecular mechanisms. These include gene